We start with a diploid cell, which later becomes a haploid cell. First, it starts with interphase. The cell grows larger in preparation for prophase 1. The pink is the DNA you get from your mom, and the blue is from dad. During S phase, the chromosomes replicate.
Next is prophase 1, where we start with 4 homologous chromosomes. The nuclear envelope begins to break down and spindle fibers begin to form. The centrosomes move to opposite poles and the chromosomes start to move closer together and overlap at the chiasmata, where crossing over occurs. Crossing over is very important for genetic variation.
Metaphase. This is when the meiotic spindle fibers fully develop and attach to the kinetochore of each chromosome. They are randomly aligned at the metaphasal plate. This shows Mendel's law of independent assortment, which further provides genetic variation.
This leads to independent assortment as each chromosome randomly links, aiding in random combinations of chromosomes in each daughter cells.
Anaphase. Pairs of homologous chromosomes separate as the meiotic spindle fibers pull them towards opposite poles of the cell. Sister chromatids are still attached, and the cell begins to stretch to prepare for cell division.
Telophase is where the meiotic spindle fibers break down. A cleavage furrow begins to form in animal cells, or in plant cells, a cell plate will form.
Cytokinesis then splits the cytoplasms into 2 separate cells. This is when it becomes haploid.
No! Is it over? No!
No! While mitosis and meiosis are similar, they have many differences. For one, crossing over, as well as randomly orienting themselves on the metaphase plate to be unique, while meiosis is genetically the same. Another difference between the two is that it divides twice, in meiosis 2.
Meiosis 1 versus meiosis 2. In meiosis 1, crossing over occurs during prophase 1. Homologous chromosomes separate and interphase happens. This results in 2 daughter cells. Meiosis 2 is when sister chromatids separate. This results in 4 genetically unique haploid cells and ends with half the number of chromosomes as parent cell.
In prophase 2, chromosomes recondense, meiotic spindles begin to form, and the nuclear envelope once again breaks down. But unlike prophase 1, crossing over does not occur.
In metaphase 2, sister chromatids, not homologous chromosomes, line up at the metaphasal plate.
Anaphase 2. Proteins at the centromere break down and the sister chromatids break apart and move to the opposite poles.
In telophase 2, a cleavage furrow is formed and spindle fibers break down. The nuclear envelope is redeveloped.
In cytokinesis 2, the cytoplasm separates, resulting in 4 genetically unique haploid daughter cells.
Overview. Meiosis begins with 1 diploid parent cell with 4 chromosomes, and ends with 4 genetically unique haploid daughter cells with 2 chromosomes each.